Okay, so so good day everyone. So now is already 9 o'clock. Okay, so let me check the screen. Okay, so now is already 9 o'clock, so we will start the class now. Okay, so today class is I changed the date because this Saturday I am unavailable, so I changed the date. It become today lah. So so today we will continue chapter 1. So last week we already cover water and also carbohydrate, right? So today we will cover another 2 biological molecule, which are lipid and also protein. Okay, so so now we proceed to lipid first. So lipid, I think everyone should know, right? What is lipid? Maybe what is lipid? So if we sorry, wait Okay, so we proceed to lipid. Okay, so you need to know what is lipid. So lipid, if we according to the atom, the organic chemistry, so normally lipid containing carbon atom, hydrogen atom, oxygen atom, nitrogen, and also phosphorus atom. The difference between water and also carbohydrate, lipid does not have the fixed ratio one. There are no fixed ratio for the lipid. So there are many, you can say there are many manner to control the lipid. So lipid containing carbon, hydrogen, oxygen, nitrogen, and phosphorus. So normally if you remember about the ester, right, you still remember the SPM that you learn about the hydrocarbon chain, that chapter, you learn about ester, right? So ester actually is one type of lipid too. One type of lipid. So normally lipid we are classified into either these it is the saturated fat or unsaturated fat, and then the common functional group are ester. So ester. So ester actually is if we change to common name, lipid lah. Because lipid containing the ester functional group. So we still remember what is ester functional group. It is a combination of carboxylic group and also hydroxyl group, right? You still remember hydroxyl group and also carboxyl group to form the ester. Okay. So normally, lipid property has these all properties, including they are maybe liquid or maybe solid form at room temperature. So you need to see lah what type of lipid, what type of component. So like saturated fatty acid and also unsaturated fatty acid, right? This one later we will see it. And also pure fat and oil are normally colorless, odorless, and tasteless. If we consider pure, you cannot see that that you see in your kitchen food oil, right? That you use the oil during cooking, or you using the fried chicken, fried rice, all that. During the cooking, your oil is already been processed, already got additional chemical or additional substance, additional organic nutrient, all like that. So it can change to yellow color that you see in the kitchen. But actually, if you want to see pure fat, totally pure, 100% pure, there are actually colorless one. They are colorless, similar like a water one, and also odorless and also tasteless. Actually, lipid fat does not have taste one. Yeah, right. If you only drink test the oil, the cooking oil, any cooking oil, you will find that there actually no taste. Actually no taste one, no taste one. So that is why you cannot drink oil. If you drink oil, actually no such thing because it is tasteless. And also there are one of the property is there are energy rich organic molecule. It is highly energy rich compared to other type of biological molecule, especially carbohydrate. Although carbohydrate. And also another thing is it is insoluble in water. Compared to carbohydrate, if you see monosaccharide and disaccharide, normally can dissolve in water one. They soluble in water, except polysaccharide. Polysaccharide cannot soluble in water. But any lipid is insoluble in water. But they are soluble in organic solvent. So like alcohol, acetone, benzene, all these, especially alcohol. If you pour the oil into the alcohol, oil will disappear. You will not see the oil in the alcohol liquid one. Compared to water, if you pour the oil into the water, you will see two layer, right? Oil will be at the above layer. Why? Because oil cannot soluble in water. And another thing is there are no ion charge. Compared to no ion charge. Okay, so and then this one is the general function of the lipid. So normally the main function is energy storage. I already mentioned it is the rich organic molecule, right? Organic molecule, so it will play major role in the energy storage. And another function is to regulate hormone, because some of the hormone actually is considered as lipid. So for example, like estrogen, testosterone, and also cortisol. These all hormones are lipid. These all hormones are lipid. They are actually considered as lipid, under category of lipid. So they are regulate hormones. So meaning that they are part of the production to produce this kind of hormone. And also transmit nerve impulse. Lipid can generate myelin sheath, which can speed up the rate of impulse transmit. This one later you will see in chapter 2 about the nervous tissue, but I think this one you also learn in SPM but I don't know you still remember or not. The nervous tissue, you know, like animal tissue we contain epithelium tissue, nervous tissue, connective tissue, all that, right? Now one of the tissue is nerve tissue. Myelin sheath is one of the component that on the neuron that can speed up the rate of impulse transmission. Another one is cushion vital organ. It provides a cushion to help to protect our organ. So normally this is just it's just like a layer. We can say that it is simply like a layer to protect our organ, right? Our internal organ, especially in our stomach. Because you know that in our stomach area, we do not have bone. We only containing skin and also adipose tissue only. Which is fat only. This fat actually protect our internal organ, especially stomach, your small intestine, large intestine, pancreas, all that. Because at that part, no bone. There are no bone to protect us. Compared to lung, if you see the lung we containing rib cage, right? Rib cage can protect our lung and also our heart. But in our stomach, with the organ within the stomach, there are no bone to protect. So fat tissue can provide the cushion to protect our organ. So it is good, but if too much, then it is not good Okay, and maintenance of temperature, of course, if you angular temperature, of course, because fat act as insulator. This one is very easy. If you go to aircon world or you go to the very cold country, you can see that the people which is very fat, they are not easily to get cold compared to the people which is very kurus which is very thin or fleet. So that people will be easily to get cold. Mereka rasa sangat sejuk punya. Tapi berbanding kepada orang yang gemuk sikit, mereka tak akan rasa sejuk walaupun dalam temperature yang sama. Okay. and then this one is transport fat soluble protein, okay, and then intra and intercellular signaling event. So normally this one can be act as the receptor lah. And final one is serve as structural component of biological membrane. Yeah, one of the example is phospholipid lah. Right, phospholipid. You know that although not entering chapter 3, but in SPM you know that you got one chapter is learning about the plasma membrane, right? You know that the major component of the plasma membrane is phospholipid. So why it is called a the terms already mentioned, right? Phospholipid already got lipid, so the major component is lipid. Yeah, later we will see the structure of the phospholipid too. Okay, and then now we consider to the structure of lipid. Okay, if in lipid there are two general structure, two main structure. Either it is open chain structure that you see in triglyceride, like fatty acid, or this is the open chain structure, hydrocarbon chain one. But there are also fused ring structure like this. This one is also considered fat. They are also not considered, also considered as lipid, but it is in fused ring structure. You can see this in the cycloalkane, or benzene structure. Okay, benzene structure. So this one is we call as fused ring compound. So normally the open chain one is insoluble in water and soluble in organic solvent, as mentioned before. And another one is amphiteric hydrocarbon and also useful for long-term energy. So yeah, so we will see it one by one. So this one is the most famous lah. Everyone should know already. Trialcyl. This one, another name is triglyceride, and common name is fat lah. Triglyceride. But similar lah. Trialcyl or triglyceride same. Okay, now we go to fatty acid first. So we go to open chain first. We go to the open chain fatty acid. So I think fatty acid, you know that if you want to form the triglyceride, you need 3 fatty acid and also 1 glycerol. So we see the glycerol first. So normally glycerol, you can see that it is the 3 carbon alcohol compound that containing 3 hydroxyl group. So you can see that there are 3 carbon. So normally if you still remember, if 3 carbon, what is that? Methane, ethane, right? Methane, ethane, then propane, right? Propane, and then butane, and then pentane, all that, right? That you already learned. So this one actually, if there are no hydroxyl group, it is pentane. But now there are 3 hydroxyl groups, so this is one of the alcohol lah. You can consider as alcohol. So normally glycerol is the 3 carbon alcohol that containing 3 hydroxyl groups. So like that lah. And then another one is fatty acid. Okay, so fatty acid is long hydrocarbon chain that containing carboxyl group. So you can see this general structure. C carboxyl group is C double bond OOH, right? COOH, right? That is that you write in your SPM, right? COOH, but actually is in the C double bond O and hydroxyl group. So normally R will be the hydrocarbon chain. This is a very long hydrocarbon chain. So in fatty acid, there are no fixed hydrocarbon chain, but different type of fatty acid will have different type of carbon chain. So normally the hydrocarbon chain, the number of carbon is normally is 9 or even up to 10 also have. Yeah, also more than 10 carbon in one hydrocarbon chain. So although COOH, the hydroxyl group is polar, but due to the hydrocarbon chain is very long, and this is a non-polar molecule. So this structure give that fat cannot soluble in water. Because due to this long hydrocarbon chain, they already cancel the effect of the polar group. So that is why that fat cannot soluble in water, although they are containing the polar functional group. However, due to the non-polar interaction is too high, already cover up the polar interaction, so that the fatty acid, the fat cannot soluble in water. And then this one is function of fatty acid. Okay. So one of the function is the building block of the lipid lah. Lipid, modification of protein, regulation of membrane unity, serve as hormone and also intracellular message, and final one is energy storage lah. And then as you know that there are two types of fatty acid: saturated fatty acid and also unsaturated fatty acid. So the key difference is the double bond. Saturated fatty acid is all of the carbon inside the hydrocarbon chain are single bond. There are no double bond or triple bond in each other one. So you see this structure totally single bond. There are no double bond except the carboxyl group. But we consider as a hydrocarbon chain, there are no double bond or triple bond. But unsaturated fatty acid is the acid that the hydrocarbon chain containing at least one double bond, at least one. Not necessarily only one, but they are at least one. Maybe one, maybe two, maybe three, maybe four, like that. They should at least one double bond group. So you can see that in this structure, unsaturated fatty acid, they containing double bond at here. And then in unsaturated fatty acid, as I mentioned before, they are either containing one double bond or they are containing more than one double bond. So normally, if there are only one double bond in the fatty acid, we call as monounsaturated lah. Another one is when there are more than one double bond, maybe two, maybe three, maybe four, more than two lah, or they are considered as polyunsaturated fatty acid. And also arrangement of unsaturated fatty acid is different compared to the saturated fatty acid. Saturated fatty acid is totally straight chain only. They are totally straight chain. So saturated because they are single bond. All the carbon is filled up with the hydrogen atom. So they will produce the only straight chain. Garbage to lose the. But in unsaturated fatty acid, due to the presence of double bond, they can be either band shape or straight chain. Band chain or straight chain. Based on the structural form lah. Either this is the cis form or trans form. So if the hydrogen the structure is cis form meaning that the hydrogen atom is located to each other at the same side. You see this double bond, right? The hydrogen carbon are totally at the same side, either at upper part or bottom part. One of them but if the hydrogen carbon are arranged to each other at the same side, so this one is cis form, they will produce the band shape. This one is called kin structure. And then this one is very important in terms of the plasma membrane. Later you will see it in plasma membrane. This structure is very one of the function because it can prevent the unsaturated fat packaging together closely to solidify at room temperature. So that is one of the reason why unsaturated fatty acid is liquid form in room temperature. One of the reason. But if the unsaturated fatty acid is in the transform, meaning that the hydrogen atom are different side at each other, so it will like the saturated fatty acid it will be straight chain. Fatty acid also classified into either essential or non-essential lah. So normally essential amino fatty acid will be fatty acid that must be obtained from diet lah. We cannot synthesize it. But non-essential can be synthesized in our body. Our body can synthesize this one. but essential cannot. You totally need to consume out, similar to the protein lah, amino acid. Amino acid also have essential and also non-essential. But essential is you must take from diet. So what normally is example is the plant oil lah, like omega-3, omega-6, all that, or plant oil, fish oil, all that normally is you need to obtain from the outside. Your plant, our body cell cannot produce this kind of fatty acid. Okay, so this is a general difference of the fatty acid between unsaturated and also unsaturated. So while the major difference is number type of bond lah. Saturated, no double bond, only have single bond. But unsaturated, they are containing at least one double bond. Due to this, so that your number of hydrogen atom for saturated will be have higher number lah. It is maximum number lah. But unsaturated, because of double bond, so that it containing fewer hydrogen atom. And the melting point for saturated is high, unsaturated is low lah. And another key difference, I think everyone know, saturated is solid form, and unsaturated is liquid form. So another sources normally saturated is animal fat lah. Any kind of animal fat, including human, normally is saturated. So, but another one is plant oil or fish oil. They are unsaturated. So normally olive oil lah. Another one, effect in humans. So normally we know that saturated fatty acid is bad fatty acid, right? It is the bad fatty acid. So normally if you consume high amount of saturated fatty acid, you will get the cardiovascular disease lah. But unsaturated fatty acid is good for health, but not necessarily say that you eat many unsaturated fatty acid is good. No, but if you compare to saturated in the same amount, unsaturated is more healthy lah. That is why that many people nowadays that focus on health, they use plant oil rather than animal oil lah. Especially in house cooking. But in outside, normally they are still using the plant. They are still using the animal oil. But some of the restaurant, especially at the very high level one, normally they will already change to plant oil. And also effect of the hydrogenation. So normally saturated fatty acid is no effect lah. Unsaturated have effect. They can be converted into saturated fatty acid one. In terms of similarity, another one is insoluble in water lah. And also hydrogen and oxygen are present in the chain, involve the formation of triglyceride. Both of them can form triglyceride one, and made up of carboxylic acid that containing carboxyl group and act as an energy provider to store energy. Now we go to triglyceride. So as you already know what is glycerol, what is fatty acid, now we need to know how the fat is formed. So fat is formed, when the fat is formed, it is called as triglyceride. Another one is triacylglycerol lah, but you can write triglyceride is okay. Okay, so normally triglyceride is formed between the condensation reaction between glycerol and also fatty acid. So one glycerol react with 3 fatty acid, they will form triacylglycerol, which is triglyceride lah, one fat molecule. So if you compare to the high carbohydrate, especially carbohydrate, fat, you can see that fat is highly rich molecule. Because carbohydrate, although it maybe polysaccharide is complex, but this one fat is just a basic unit only. They already can form the complex structure like that. So this one is the condensation reaction, meaning that there are the water molecule is released out from there through the hydroxyl group of the glycerol and also carboxyl group of the fatty acid lah. So they will form the ester bond at here. So that is why that lipid is also one of the example of ester. Because this is another example of esterification that you already learned in SPM lah. So why esterification process lah? So yeah. Okay. And then also triglyceride also can lip fat can also undergo hydrolysis. You can break down the fat through to release the glycerol and also fatty acid through the hydrolysis lah. So you need the water lah. But actually normally you need to catalyst lah. Normally you need the catalyst. One of the main catalyst is lipase lah, lipase enzyme. Okay, so normally this one is the function. So normally similar to the lipid, normally energy storage, thermal insulation, soap making, and also organ protection lah. Okay, so saturated fat and unsaturated fat normally is similar to the difference between saturated fatty acid and also unsaturated fatty acid. Okay, now we go to the phospholipid. Phospholipid. So another terms name you also can consider as phosphoalkyl lah. So this is the type of lipid that has the similar structure to glyceride, but the difference is they are in terms of instead of 3 fatty acid chain, they are containing 2 fatty acid chain only, and another one is replaced to phosphate group. Yeah, so that is why that you see the phospholipid is like that. It's like that. There are only two tail at there. These 2 tail are considered as represented as the fatty acid chain. So 2 tail meaning there are 2 fatty acid chain. But there are another one is not fatty acid, is we consider as phosphate group. So if you see this diagram, yeah, this diagram. So another terms of the phospholipid is lecithin. This one is that lecithin. So this is the basic unit if you want to form phospholipid. It's like that. Lecithin, because they are has a choline group attached to the phosphate group at here. One of the example is due to the phosphate group. They are the phosphate group at here. So this structure is called as lecithin. They are soluble, water soluble, emulsify, and membrane constituent. So this one is the difference is they are partially soluble in water one. Compared to the fatty acid, the fat triglyceride is totally cannot dissolve in water. They cannot soluble in water one. But this one is partially. Why? Because of this one is polar group. And these two, of course, non-polar. So this one is the difference. So you can see that phospholipid, they're containing one phosphate group at here. Phosphate group at here. So you will see the triglyceride, there are no phosphate group. 3 of them are fatty acid. So phospholipid only containing 2 fatty acid. So that is why that if you see the phospholipid, they will have hydrophilic head and also hydrophobic tail. Hydrophilic head meaning is soluble in water, right? This one is face to the water one. And another one is hydrophobic tail. This one is repel water. Repel water one. So that is why that you see the phospholipid arrangement is like that. It's like that. At the outside of the surface is containing water molecule. Fatty acid does not like it, so the hydrophilic head will exposed to that lah. Similar to the lower part, why is reversible? The hydrophilic head is at the face into the cell, because inside the cell they containing water. So hydrophobic tail, the fatty acid is not like water, they repel water, so the hydrophilic head will expose to that water lah. So normally, if you see the behavior of phospholipid in water, as I mentioned before, they are philic and also phobic. So normally, phospholipid is, you can say that it is partially soluble in water. They are not totally soluble, but they are partially soluble. The hydrogen ion dissociate from phosphate group. This causes the phosphate to be the negatively charged. There are the reaction. If you still, that reaction occur when you dissolve in water lah. So that is why that when you arrange the phospholipid in water, they arrange at like this in form of phospholipid bilayer. That is why that there are 2 layer, bilayer, 2 layer at here. Why not just only one layer? Because if there are only one layer, inside the cell also containing water. I already mentioned fatty acid is not like water. They are repelling water one. They repel water. So they cannot expose to water, so that they were arranged in terms of phospholipid bilayer. Another one is they are in terms of the micelle, also membrane bulk vesicle like that. Okay. Yeah, so you see in plasma membrane, for hydrophilic head will face to the cytoplasm and also extracellular fluid, while your hydrophobic tail will hide inside the layer of the hydrophobic center. Okay, now we already finished up the open chain lipid. These are all the open chain lipid. So there are two open chain lipid that you need to learn: fat, triglyceride, and also phospholipid. Phospholipid, another term is lecithin. Okay, now we go to the fused ring structure. This is the fused ring structure. We call as steroid. Steroid. I think everyone know what it. Maybe heard about it. What is steroid? So normally, if you go to hospital, some of the disease there, doctor will tell you, right, or when your immune system is weak or something disease, they will inject steroid to you. So what is steroid? So normally steroid is the fused ring structure of the lipid. So there are many examples of the steroid. So these are the one of the example, 5 example, common example that we normally see in our life. So all of us have this hormone except the sex hormone. So all the sex hormone are under steroid. So you can see progesterone, estrogen, also testosterone. So progesterone and estrogen, both of them are female sexual hormone, right? That play the major role in menstruation, and testosterone is a key hormone for male. And also there are aldosterone. This one is play the role in homeostasis. And another one, the most famous one, cholesterol. Right. So you can see in our syllabus, if we refer to MPM syllabus, the main focus is cholesterol. The other you don't need to know the structure, but except cholesterol, you need to remember the structure. You must remember. You need to remember or how many chain. You can see there are the 1, 2, 3 hexagon structure and also 1 pentagon structure. And the number 2 hexagon structure, they are containing the double bond at here, and you need also know that there are the hydroxy group, there are the methyl group, methyl chain, and there are the hydrocarbon chain at here. You need to memorize it. Why? One of the year in essay, MPM asks you to draw the structure of cholesterol. With the aid of diagram, the question is like that. With the aid of diagram diagram, explain the structure of cholesterol. They need you to draw a cholesterol. So you must memorize it lah. Others you don't need to do. Because in our syllabus they only mention cholesterol. The other I just let you know only. Aldosterone, progesterone, estrogen, and also testosterone are the example of the steroid. But cholesterol is you must remember. Okay, so this is just a repeat. Cholesterol. So the cholesterol structure is, there are 4 hydrocarbon chain. 1, 2, 3, 4. There are 4 hydrocarbon fused chain, fused ring. Is with the hydrophobic tail at one end of the hydrophilic region, and also OH group at another end. You need to know that this one is hydrophilic region and hydrophilic region, and this one is hydrophobic region. You need to know hydrophobic and hydrophilic. You need to label. You need to label too. To got marks. If you not label, you just draw only, no marks, or maybe 1 marks only lah. You cannot get full marks. And the importance of cholesterol in health normally is this. So I think this one everyone should know lah. Why cholesterol is very important. So normally one of the function is the formation of cell membrane to maintain our healthy skin. Also, require production of male and female sexual hormone. Why? As you know that cholesterol is steroid, and you see the other structure of steroid almost similar to cholesterol. You can see almost similar. The hydrocarbon chain is totally same. There are 3 hexagon and also 1 pentagon, totally same. The difference is that the functional group only. Maybe the functional group, you see the testosterone, the function there. This part is not hydrophobic tail. This one is hydroxyl group, and this one is carbonyl group, and this one you can see. So meaning that, an example in simply way, cholesterol can be undergoes the certain chemical reaction, certain enzymatic reaction to convert into these all things. So that is why all the function is say they can produce male and sexual female hormone. Also formation of testosterone. Of course. I already mentioned at here. You can see structure totally same. The key, the mother carbon is totally same. The difference is just a side chain only. You can see the difference of the cholesterol just at here. This one is the carbonyl group. This one is hydroxyl group, right? Hydroxyl group, carbonyl group. They can change, and you can see the carbon bond. While the carbon bond is at here, and while the carbon bond is at here, they can change one. And this one is same, and you can see the end of the here is also changed. But I mentioned, undergoes certain enzymatic reaction, cholesterol can change into aldosterone, similar to progesterone, testosterone, and also estrogen. Also formation of vitamin D in skin, and also formation of bile salt, and maintain stability and permeability of plasma membrane, and metabolize lipid, and also dissolve lipid soluble vitamin. So all this about the cholesterol in health. So I think that's all about the lipid. So any questions so far? Okay, no question. Okay, very good. Okay, if no question, next we will go through the protein. Okay, protein, I need to get you know that protein is one of the famous topic that normally MPM like to come out. Wait a minute. Wait a minute. I need to settle something first. You guys can take a rest first.没有啊，在翻呢，该 settle 那个 slide。哎呦！不是暂停录制啊，暂停学 Okay, now we proceed to the protein. Okay, as I mentioned, protein is one of the famous topic. Mostly MPM come out, especially in whatever essay question, structure question. The most topic in chapter 1. Normally in chapter 1, if MPM one come out topic in chapter 1, normally is protein. Compared to other type of biological molecule, protein is the most famous topic to be coming out rapidly in exam question, real exam question. So this topic is quite long. It's quite long topic, and most important I can say this chapter. But not say that other chapter is not important. Also important but this chapter is one of the topic that MPM always coming out in essay. Okay, so protein. Okay, so I think I don't need to talk much. Protein, as you guys know that the basic function, the basic unit of protein is amino acid, right? Amino acid. So this is the structure of the amino acid. You can see that basic amino acid structure is they containing the carbon at the center. At the center, carbon at the center, and they're containing two groups. They are containing amino group, and also carboxyl group. So that is why that it called as amino acid. Amino, this term is coming from amino group. Acid is come from the carboxylic acid. Carboxylic acid, right? Because, right, okay. So amino group is NH3 lah, amino, right? Amino normally is NH2, NH3, sorry, and carboxyl is COOH. OH. So there are one amino group at here, NH2 bonded with at the center, and also carboxyl group bonded at the center. And there are the R group, which is a side chain. And we know that there are total in real world, whatever organism, there are total 20. There are only 20 amino acid in the world. Only 20. There are no more than 20. Only 20. Compared to carbohydrate, compared to lipid, there are more than 20 lah, maybe more than 20. But amino acid, there are only 20 amino acid only. 20 only. There are only 20 only. So so meaning that there are 20 different R. There are 20 different R. Different 20 R lah, we can say that. Okay, so this one basic structure, you must know it. You need to know it, how to draw it, like that lah. COOH and NH2, and there are the H at the bottom, and also R group at here. So what is protein? So I mentioned monomer is amino acid, and there are 20 different type of amino acid and can be combined to form a protein. And so protein, why protein is very complex? Because it is have a larger range of the function. There are many functions. So what can you imagine what function? What function? You will still remember what function that you learn from SPM. One of the key component is enzyme. You know, like enzyme is protein. Enzyme is protein. And then beside enzyme, antibody. Antibody also one type of protein. And apart from antibody, what another? Your plasma membrane, right? Your plasma membrane, you also containing protein channel, right? Core protein, carrier protein, all that. And also another one that you already learned. Hormone. Protein also is a hormone. So the function of the protein is very large. So that is why that protein is very important. One of the component is very important to the body. Okay. So this one is all the 20 types of amino acid that you need to know it. According to MPN syllabus, you should memorize all. But I think you are human, right? No one can memorize. Even me too, I cannot memorize. Even me, I cannot memorize too. I only can memorize one this glycine only, because glycine is the most basic one. It is the most basic one. But I only can tell you that you can choose any type of the amino acid. Why I say you need to memorize all? Because one of the year MBM, they coming out in structure question, they just coming out this type of structure, and then you need to name it. Any type, or if no mistaken, 3 type. They coming out glycine, alanine, and another one is I already forget. Any 3 type. They give the diagram lah, these 3 diagram, and then they say PQR. You need to know lah what is P, what is Q, what is R. And then entire question is totally based on these 3 amino acid. So if you totally don't know what is that, what are the amino acid, you totally cannot score that structure question. So yeah, so that is mean that in MPN syllabus you need to know all. You should know all lah, but yeah, you all are. So I cannot. In real world, I cannot ask you to memorize all lah. This is unlogic. I memorize all, but try as much as you can lah. Okay. So yeah, as I mentioned, there are only 2 also essential, also non-essential. So you know that essential, there are 9 essential amino acid and also 11 non-essential amino acid. So what are the 9 amino acid? Essential amino acid, this 9 lah. And also what are the 11? This 11 lah. So the main difference is that essential is you must obtain from diet. Your body cannot produce one. But non-essential, yeah, your body can produce this. Okay. So this one is a function as I mentioned, structural function, and also contractical function, storage, protein is also storage, and also defenses, as I mentioned, antibody, transport, one of the transport, hemoglobin, messenger, hormone, and also enzyme, all that. So you can see that actually whole protein has the very large range of the function. Okay, so backbone of the amino acid. So as I mentioned before, backbone is R group. So as I mentioned, there are 20 types of backbone in amino acid it contribute to 20 types of amino acid. Okay, so this one is the central carbon. You can see this is the central carbon. It is located at the center, and there are carboxyl group, carboxylic acid group, and carboxyl group, hydrogen atom, R group, and also amino group are bonded to each other to form the alpha carbon. So normally this one is we call as alpha carbon since amino acid is at the center. And amino group, as I mentioned, is NH2 and carboxyl group is this COOH, C terminal. When you talk about the N terminal and C terminal, N terminal is the amino group, and C terminal is the carboxyl group of the amino acid. Another one, this one is very important. This is what we call as deuterion. This one is that when the amino acid is inside the water, when is immersed in the water, soluble in water, I can say when amino acid is inside the water, they will form deuterion structure, which is that they will accept and also receive electron all together. Because they containing amino group and also carboxyl group. We know that amino is basic, acid and base. You still remember your SPM chemistry. This one is more focused on chemistry part. You still remember about your SPM chemistry. We know that in acid and base reaction, acid is hydrogen ion, right? Base is hydroxyl group. While the hydroxyl OH. And you know that amino, ammonia, all that is basic. And carboxylic acid, any acid is acid right. So if you still remember, acid is release electron, release proton, release hydrogen. And another one is basic, accept proton. They accept proton. They can accept hydrogen ion. One of the theory. This one is chemistry theory, the basic chemistry theory for the acid and base. So in amino acid, due to it containing both basic group and also acidic group, so that they can form internal transfer of a hydrogen ion from the carboxyl group to the nitrogen amino group. So it is very tricky, meaning that where it is released water, where it is in the water, acid substance in zero situation, acid substance they will release hydrogen ion. So as your carboxyl group must release hydrogen ion. Okay, and then your amino group in zero situation, if they are only containing amino group or basic group, when they is immersed in water, they need to receive hydrogen ion. So this one will create a balance that your hydrogen ion for the carboxyl group will go to the amino group, so that they will both go to each other. And then we know that when acid that release the carboxylic acid that release hydrogen ion, they will form carboxyl group, negatively charged, and amino they will form ammonium ion which is positively charged. And this type of structure is we call as deuterion. And due to this structure, they are also can be act as the buffer solution. Amino acid can be act as the buffer, so that when you add little acid or base is added, there are no, the pH of the solution will remain unchanged. This is the function of the buffer. Later in chemistry, just want to let you know, in your chemistry, chapter 6, equilibrium, you will learn about more acid and base, acidic reaction and also basic reaction. When you learn about that, you will know the function of deuterium. Maybe now you are confused again, but this is very simple theory only. But in chemistry, they are more complicated lah. In chemistry, you are also learning about the buffer too. They are the definition of the buffer in chemistry. So actually, the concept is still same, totally same. But in bio, I just explain in simple only. But in chemistry, your chemistry teacher will explain more detail lah. But in buffer, when you recall as buffer, meaning that when you add little acid or little base, meaning that it's weak acid or weak base is added, the pH is still remain unchanged. There are no pH change one. The pH is still. If you consider to the normal situation, if you add water I consider as water. If you add acid, whatever weak acid or strong acid you add to it into water, when you measure the pH value is will be down. It will directly decrease one. But in this protein, in amino acid solution, when you add only weak acid or weak base, you add to it, your pH still remain unchanged one. So that is why that we call as deuterium. So this is very important, very important. I can say very important structure. One of the major property in the protein. Okay, so what is R group lah? So R group is a side chain. It is a side chain. So normally your R group is determine the characteristic of your amino acid. So amino acid not only classified into essential and non-essential amino acid. The classification of amino acid is based on the R group. Now different R group will be provide different characteristic to that amino acid. If you consider this basic structure of the amino acid, it is actually act as the neutral. This one is neutral, meaning that pH is equal to 7. If you ignore the side chain, if you assume that the side chain, the R is HR glycine. If you assume this amino acid is glycine, it is neutral, pH equal to 7. But different side chain will provide different property to this amino acid. So there are four main although amino acid can be classified in various ways, one common approach is to classify according to what the functional group of the side chain at neutral pH, either meaning that at pH equal to 7, either your amino acid is non-polar, polar but uncharged, polar, uncharged or meaning neutral, or negatively charged or positively charged. So you see this diagram, you can see there are total 1, 2, 3, 4, 5. Five main group of the amino acid: non-polar amino acid, polar amino acid, aromatic amino acid, positively charged amino acid, and also negatively charged amino acid. These 5, you need to know it. Now we will go to one by one. So as I mentioned, amino acid normally is considered 4 categories: acidic amino acid, basic amino acid, polar amino acid, and non-polar amino acid. Very important. Acidic is positively charged, acid is negatively charged, basic is positively charged, polar and also non-polar. These are very important. You need to know it, and you also need to know the amino acid too. So one of the difference is we want to compare the difference between basic and also acid. Acid is based on the side chain. So you can see acidic amino acid, you can see that one of the example is these two lah, aspartic acid and glutamic acid. Already mentioned acid. Well, why? Due to the side chain, they also containing the the carboxyl group at here. You can see they are also containing alkoxyl group at here. So when they dissolve in water, they will, as I mentioned, in acid condition, acid solution, if it is acid, they will release hydrogen ion. They release hydrogen ion so that they will produce acidic solution. So that is why that in neutral, meaning that when you immerse in water lah, pH equal to 7, this amino acid will cause your solution become acid. Your pH will drop. Because why? They're containing two negatively charged. One is the basic unit one, another one is based from the side chain. So acid producing effect will be produced. So this one is we call as acidic amino acid. They will produce acid solution. And this one is basic, meaning that they're containing ammonia group at here. So arginine, lysine, and histidine, both three of them are basic amino acid where they containing amino group. Normally they contain nitrogen atom. You can see when they are containing many nitrogen atom, you can see they containing ammonia group, amino group, ammonia, and even this aromatic one they containing high nitrogen atom. All these will provide basic amino acid, meaning that they will when they immerse in water in neutral pH, they need to accept hydrogen ion. So when they accept hydrogen ion, so that they will produce basic amino acid. But as I mentioned, you need to be alert. Well, amide group like amide group CO and H2, so normally asparagine and also glutamine are not considered as basic amino acid. So you can see this, where glutamine, you can see this glutamine are not considered as the basic amino acid. Another one is what? Arginine, lysine, histidine. Yeah, this one is positively charged. Another one, this asparagine and also glutamine, they are polar, but they are not basic. Why? Because they are amide group, C double bond O and H2. This one is not considered as basic. They cannot accept hydrogen ion one in simple, so that this one is not considered as positively charged. They are polar amino acid. Okay. So this one will cause the amino acid, this property, the deuterium property will cause that either your amino acid is in acidic or basic solution at the similar situation, meaning that at pH equal to 7 So meaning that when you are equal to pH equal to 7, they will form deuterium. But if you increase your hydrogen ion, you increase your hydrogen ion or decrease your hydrogen ion, so you can see that at high pH, meaning that your hydrogen ion is very low, deprotonation will be happen. Deprotonation meaning that they will only release the hydrogen ion only. And if your acidic condition is high, your surrounding if your pH value initial pH value is very low, is highly acidic, meaning that protonation only happen. COOH is still maintained. The hydrogen ion will not be released, but your amino group still accept the hydrogen. That is why we call as protonation. Deprotonation is release hydrogen ion. So this one is the concept of equilibrium later that you will learn in chemistry lah. Okay, so this is the protonation lah. So normally when your hydrogen ion is very high, meaning that your pH is dropped, so that your carboxyl group only will accept the proton to become uncharged COOH. So your overall will be the positively charged. So compared to deprotonation, where your hydrogen ion is very low, pH is high, meaning that in very acidic condition, so that your carboxyl group will lose the proton to become CO negative, so that your overall will become negatively charged. And then this one is the differences between polar and also non-polar amino acid. So normally polar amino acid normally is hydrophilic, they can soluble in water, but non-polar amino acid they cannot soluble in water. This one right, polar can soluble, non-polar cannot soluble. This is a general concept lah. And also normally polar amino acid, your R side chain normally is partial positive, either partial positive or either partial negative, so that charge group. But in non-polar, normally they are neutral, totally neutral. Normally they are neutral. There are no group, no charge group at there. No charge group. Does not carry any charge group. So that polar amino acid normally can found on the surface of the protein, but this one is found interior part of your protein. You know protein. I already mentioned protein is the combination of many of amino acid. So polar amino acid you can find at the surface, and non-polar is found on the interior part. And normally polar amino acid you can soluble in aqueous solution lah, like water, but non-polar is soluble in organic solution like alcohol. So the all the polar amino acid including tyrosine, serine, you can see they are hydroxyl group 1. Hydroxyl group, they are polar group 1. Hydroxyl group, and they got carbonyl group, amide group, and also sulfur group at here. Okay. And then if you see the non-polar, normally is totally hydrocarbon chain 1. Like glycine is the basic unit lah. Glycine and alanine you should remember lah. This one is most easy one lah. Glycine is the general one, and alanine is only containing one methyl chain. Valine is isomethyl, and then leucine, isoleucine, proline, tryptophan, phenylalanine, also methionine. All these are considered as non-polar amino acid. You can see that they are normally is containing hydrocarbon chain. Okay, so that's all about the amino acid. Okay, next we go to the peptide bond. Okay, so we already learn about the amino acid. Now we go through the peptide bond. So what is peptide bond? So peptide bond is the amide type of covalent bond that link two consecutive alpha amino acid from C1 of one amino acid and N2 of another. So if I see you the diagram, more easy like this. So these two are glycine. This glycine 1 and glycine 2. So if you want to form peptide bond, one part of the hydroxyl group and another part of the another amino acid of the amino group will release one hydrogen molecule, so that OH group, hydroxyl group from the carboxylic acid, and also one hydrogen atom from the amino group will be released out to form water molecule, so that this is condensation process. They will form peptide bond. So they are the amide covalent bond at here, CN. The C bonded with N. So this C is one amino acid, one glycine, and bonded with another glycine. So this one is peptide bond through the condensation process, where we can release water, release water. Okay, so dipeptide is the structure. If there are two amino acids joined together, then this structure we call as dipeptide lah. Okay. And then this one is one of the important. So when there are many condensation process, if condensation process is rapidly occur, so they will form the protein molecule with different structural level. So either they form primary structure, secondary structure, tertiary structure, or quaternary structure. So if at the higher level of structure, the more complicated of the protein structure, and also the higher number of amino acid. So in primary structure, very simple. You see this diagram. Primary structure is just a single polypeptide chain. Polypeptide chain is that when there are many amino acids bonded together through the condensation process. They are bonded together. They will form primary structure, which is there are only single polypeptide chain only, like this. Only single polypeptide chain. So normally this one is the main structure is covalent bond. They are only covalent bonded at here. So you see this one of the example insulin lah. They are only single polypeptide chain only. Okay. Another one, secondary structure. Okay, so this one, secondary structure, is that there are one bond which we call as hydrogen bond. Yeah, that you already learned in chapter 1. But this hydrogen bond is not formed in water. This hydrogen bond is formed if within the same polypeptide chain, there are hydrogen bond can form. If you still remember in water, how hydrogen bond can form? Partially negative. If there are the one water molecule and bonded to different water molecule through the partially positive hydrogen and partially negative oxygen, right? And different water molecule, then they join together, they will form a hydrogen bond. Similar to the protein too, but in protein they are not different protein lah. They're just that different location of amino acid. In the different location of the amino acid, if there are partial negative oxygen and also partial negative hydrogen, they're able to form hydrogen bond so that they can coil or fold to the polypeptide chain, so that you can see the polypeptide chain is like that. Rather than this one is just a basic unit only lah. You just join like that only. But this one is not join. They not only join, but the structure is coiled to each other. They form coil structure. You can see they switch, right? Macam spring macam ni, and you can see ini macam kertas. Yes, this is what we call as folded. So there are two common type of secondary structure. Either is alpha helix is like that. This one is the helix structure, coiling structure like that, or beta pleated sheet structure. This one like that, like a paper. You can see it is like a paper one. Both of them are held by hydrogen bond. Why they can form like that? Due to hydrogen bond. Hydrogen bond can make the structure like that. The key component is hydrogen bond. If no hydrogen bond, they will just form like that only, like this only. Just a covalent bond, they join together, only like that. But this one, due to the presence of hydrogen bond, so that they will form helix structure and also beta pleated sheet structure. So this one is how alpha helix structure. So it is a right-hander helical structure, and each helix contains 3.6 amino acid residue. This one is average. So normally your C double bond or group of each amino acid is hydrogen bonded with NH group, meaning amino group of another amino acid, which is situated 4 amino acid at here. So meaning that you see here, when they are the amino acid at here, and there are another amino acid here, so that they can form hydrogen bond at here. The one of the O group and also one of the N group is can form hydrogen bond at here. So like a shortcut lah. They will not only bonded like that, they will form a shortcut like that, so they can join together, tied together. They macam tarik dia ke sana, so that they conform the helix structure like this. So this one you can project outward from the twisted backbone. A beta plated sheet is totally fully extended, meaning that beta strand line up side by side, forming beta plated sheet, and it is stable between also hydrogen bond. So you can see it like this, just side by side, like this. Like this, can run parallel or anti-parallel to each other. R group are alternatively projected to above and below the plane of the beta sheet. So you can see this diagram. This diagram is like compared to the alpha helix. Alpha helix is like a single polypeptide chain, and then they just squeeze at this, like this. Macam ular lah macam tu. But ini macam paper dia. Tegak-tegak, all the amino acid is straight away, and then just that they can form the macam join macam ni, and to form the hydrogen bond parallel, anti-parallel or parallel to each other like that lah. Okay, so this is the summary of the differences. You can read yourself. Okay, tertiary structure is more complicated. More complicated because this is 3D structure already. Compared to primary and secondary, both of them are still 2D. Still 2D. Like a paper only. You like a paper you see. Normally it's like a paper you see only. But this one is 3D structure. Tertiary is the 3D structure already. 3D structure already. But still only one polypeptide chain form. Primary, secondary, and tertiary, 3 of them are only one polypeptide. There are no 2 polypeptide, 3 polypeptide. Only one polypeptide. Okay. Polypeptide. So normally it's still similar to secondary structure. What happened in secondary structure, tertiary structure pun happen, still happen. But there are additional interaction occur between polar, non-polar, acidic, and basic are group within the polypeptide chain to contribute the 3D shape. So you see that there are total 4 interaction. Hydrogen bond is one, compared similar to secondary structure. Another one is disulfide bridge, hydrophobic interaction, and also ionic bond. These 3. They can form these 3, another 3. So disulfide bridge, as you see that in amino acid, certain amino acid they containing sulfur. Right, in the side chain, you still remember the polar amino acid and also non-polar one. Both of them, some amino acid can containing sulfur one. One of the example is methionine methionine containing sulfur. Okay, so they can form the disulfide bridge to each other. This one is one interaction, and another is hydrophobic interaction between the non-polar and non-polar group. You can see this. You can see this one, if not mistaken, is valine, right? Valine, valine. Right, valine. If not mistaken, valine. This one is the hydrocarbon chain. When they are hydrocarbon chain to each other, they can form hydrophobic interaction like this. This one we call as hydrophobic. Why hydrophobic? Both of them are hydrophobic, so they can form the weak interaction like Van der Waals force. For very weak Van der Waals force to form we call as hydrophobic interaction. And another one is ionic bond. They can form also ionic bond at here. If they are the different charge. If they are the positively charged and also negatively charged amino acid, the side chain, all of them are side chain. You can see all of them are the side chain. Where the side chain of the amino acid is either positive or negative to each other, they can form ionic bond. So you see the interaction between an ion and cation of the R side in the polypeptide. Hydrophobic is when there are 2 non-polar to form. A hydrogen bond, then there are the hydrogen atom from one R side and another one is another R side. A disulfide bond or disulfide bridge is when there are the cysteine amino acid that can form SH group, SS bond, sulfur bond. So these are 4 interaction contribute to let it can form 3D structure. And last one is quaternary structure. Okay, quaternary structure is also 3D. There is also 3D, but highly complicated. Why? Because it is containing made up of many polypeptide chain. Compared to tertiary only one polypeptide. One polypeptide they already can form 3D. But this one is containing 2 or 3 or 4, at least 2 polypeptide, so that they can form we call as quaternary structure. And then the subunit may be same or may be different, meaning that the polypeptide chain can be totally same, meaning that A chain and B chain, the sequence of the amino acid are totally same, or they can different, meaning that the A chain and B chain are totally different, or there are something different at there. Maybe A chain containing 100 amino acid, B chain containing only 60 amino acid, like that lah. And then all the interaction in the tertiary structure still happen in quaternary structure. The difference is that they contain more than one polypeptide chains. So one of the major example is hemoglobin. So you can see this structure, this hemoglobin. Hemoglobin containing 4 polypeptide chains, 2 alpha chain and also 2 beta chains. beta chain, 2 alpha and 2 beta. So you can see these, the lower alpha, the upper one beta lah. Okay, bivalent group B is one of the famous group favorite example lah. Okay, next we go through the denaturation and also renaturation. Okay, we already cover all the basic of the amino protein, the level structure. Okay, now if we know that protein, if you want to break down, is very easy. If you want to alter the shape of the protein, it's very easy. It is not just that hydrolyze. Hydrolysis is just if you want to break down peptide bond. But if you want to break down peptide bond, it's not easy. Breaking up down the peptide bond is not easy. But if you want to break down the 3D structure of the protein, like this 3D structure of this, or you can want to break down 3D structure like this, it's very easy. Why? You need to know that the interaction, all these interactions are very weak interaction only. Compared to a covalent bond, at here the covalent bond, the peptide bond between the amino acid, this one is strong. If you want to break down it, it's still hard, it's quite hard. But if you want to break down, especially hydrophobic interaction, disulfide break, hydrogen bond, all these three, very easy. If you only your environment change, like your temperature is too high or your temperature is too low, or you add the acidic condition, or you add a lower acidic, or you go to radioactive environment, your amino acid, your 3D structure of your protein will change, very easily to change. So this one is that we know as denaturation and renaturation. Denaturation is your protein is already denatured, meaning that your protein is altered in shape. Altered in shape meaning that your function of the protein is different, already interrupt. You already interrupt your protein function. Like quaternary, we know that there are 4 chains. If they are bonded together, but if there are something happen, when the chain is break down, or you already break down any interaction in that this, maybe already from 2D, 3D become 2D, like that, your hemoglobin cannot function already. This one we call as denaturation, and renaturation is reforming lah. But actually, in real situation, you want to reform again the protein is not very easy compared to denature. If you want to macam senang kan, nak rosakkan sesuatu benda senang je. Nak rosakkan, just pukul je, buang je, rosak. But nak repair, susah kan. Macam accident, langgar je, okay lah satu saat. Tapi awak nak repair your car, you need to take time. So similar to protein too. You want to break down the protein structure, very easy. It's just very easy. But you want to reform your protein again, very difficult. Very difficult. Okay, so normally denaturation, change in specific 3-dimension structure, especially in tertiary and quaternary protein. Normally in our real situation, especially in our body, majority of our body protein is tertiary or quaternary one. There are very less is primary and secondary. Very there. Primary and secondary normally is in the prokaryotic bacteria lah, or very small organism one. Like human complex organism or bigger animal like that, normally our protein is tertiary or quaternary. You see human. Hemoglobin is very important. If your hemoglobin is break down, is already broken, you can imagine the function lah. Okay, so the 3D structure of the protein is relative unstable. I can say unstable. Normally these two, although ionic bond, ionic bond is strong, right? Ionic bond you see strong, but this ionic bond is fat ionic bond, not ionic bond that you learn in chemistry. You know that ionic bond in chemistry is metallic bond. It is between the formation of the bond between the metal and metal one, or metal non-metal. That one is ionic bond. That is very strong. But this one is just a fat bond only. Because this is what we call it. Why you call it as ionic bond? Because of different charge only. Different charge, then you call as ionic bond. But this bond, if you compare to the chemistry, the real while the meta bond actually is still weak. It is still weak. So you don't want to confuse with the ionic bond in this and also chemistry that you learn in SPM or in your chemistry. This ionic bond is fat ionic bond. And the denaturation of enzyme, antibody, and protein or the hormone will cause your function loss. Yeah, although your amino acid sequence still same, they will not break down your amino acid sequence. They will not hydrolyze your amino acid, your peptide bond, but your peptide bond still remain. But your interactions change, your 3D structure change, your function also change. Like quaternary, like your hemoglobin. Hemoglobin, we know that play the role in carry oxygen, right? Carry oxygen also carry carbon dioxide. If your hemoglobin is breakdown, how do you carry oxygen? You cannot carry oxygen already, then you die. So it is very important. So what can cause denaturation? This either heat or radiation. When you heat heat your protein, so that is why that at very high situation, that is why human cannot survive at the high temperature surface, high temperature places at the long time, because not only you cannot tahan, not only hot, but your enzyme also cannot tahan. Your protein, your internal body, including enzyme, antibody, all that also cannot tahan. Because this one in very hot situation, you will increase the kinetic energy inside the atom, between your atom, so that your protein, your interaction between protein will be very active. If too active, hyperactive, it can break down your hydrogen bond and also ionic bond one. When it break down, 3D structure alter. And another one, strong acid and strong alkali. If your protein is exposed to strong acid or strong alkali, what happen? Additional of hydrogen ion, this one is strong acid. If strong acid is added, it will combine with carboxyl group, COO negative in amino acid to form ZOOH. So reduce hydrogen ion in alkali cause your ammonium group in amino acid to release hydrogen ion to alkali medium to form amino. So this one will cause your ionic bond is break. You know that if you want to form ionic bond is you need to have a positively charged for amino or ammonium, and also negatively charged from carboxyl, right? Then you can form ionic bond. If not, if like that, already form neutral, already form COH back, already form NH2 back, so no charge, so already form neutral, so your ionic bond are broken. There is highly stock concentration or heavy metal also can cause. So normally this one is highly stock concentration due to you can form very electropositive ion, form a strong bond with carboxyl group in amino acid. Meaning that this one is according to ECS, electrochemical series. This one, if they are highly electropositive, so normally is metal ion. If they are metal ion, like sodium ion, you know that sodium ion, potassium ion, all these, potassium ion, magnesium ion, zinc ion, copper ion, all these have highly reactive compared to carboxyl, compared to hydrogen ion, like magnesium ion, sodium ion, copper, or even zinc. All that is highly electropositive, highly reactive compared to hydrogen ion one, so that they will form bond instead with the amino group. Actually, in real situation, your COO negative, your carboxyl group should form bond with the amino group, right? In natural situation, but due to they are highly electropositive ion, highly reactive, so that your carboxyl group will not bond with the amino group. They will form with the any electropositive, so the ion, magnesium ion, zinc ion, all that. Similar negative. If they are highly electronegative, they will not bond with CO negative. They will bond with other, so like hydroxyl group or other. Also, organic solvent and also detergent will disrupt your hydrophobic interaction. So this is the cause of denaturation can be occur in your in protein. Okay, if you want to renature your protein, so normally will sometimes spontaneously refer to its original structure. So normally this one is you can provide physical and chemical environment to normal with the nature action removed. So protein functional structure remain the same and normal biological can be carried out. Okay, final we go through the classification of protein. Okay, this is the final. But this one you don't want to confuse with the level structure. That's not primary, secondary, tertiary, and quaternary. These are no classification. That one is no classification. That one is the level of structure. If the exam want you to classify protein in this topic, if exam ask you to describe the classification of protein, you write primary structure, secondary structure, or there is no classification. That one is level of structure. You need to different it. You cannot confuse it. You confuse it, then do smart. Okay, so normally in classification we are two main classification. Either you are simple protein or conjugated protein. Simple protein is that your protein only containing amino acid, But conjugated protein is contain not only amino acid, they are also containing what we call as phosphatic group, a non-protein material. Any non-protein material. One example is quaternary, hemoglobin. Hemoglobin, we know, right? They contain hem group, meaning that this one is metal ion. We know that it containing metal ion, the ferrum ion at there, fetal atom in the hemoglobin. So hem, your metal, your ferrum is phosphatic group. So they are not only containing amino acid, they are also containing the ferrum. So hemoglobin. So these are all the examples. And also glycoprotein. Glycoprotein also one of the protein. They are not only the glycoprotein, right? Glyco meaning that they are carbohydrate chain bonded to that protein. So this one, your phosphatic group is carbohydrate. And also nuclear protein, casein, or this is example. Another different classification is globular protein and also fibrous protein. So you can see that globular protein normally is the very circular shape, 3D shape, circular, 3D, spherical, round structure. So normally it is made up of any type of protein. Structure of the protein amino acid can be formed like that. So normally this one is the explanation. And fibrous protein, you will see that fibrous protein is a long and narrow and normally repeating set of amino acid. So normally the role is strength and support. Just now, this one is role as enzyme and transport. Strength and support. And production is movement, and also surface is covered in non-polar amino acid. Okay, so this is the difference between fibers and globular protein. Okay. And then this one is just a general function of the protein. So this one you can read yourself lah. I don't want to talk much. So yeah, function of protein, as I mentioned, function. One of the function is enzyme lah. So this one is one of the chapter 4. You will learn it. So I don't want to talk much about this. So chapter 4 I will talk more detail lah. Another one, as we mentioned, is we can provide protection and also support of the structure. So normally like collagen, your elastin, fibronin, all this is protect or support to your body. And movement like your actin, tubulin, myosin, cytoskeleton, all these also involved in movement. And defense, I already mentioned antibody lah. One of the antibody. Not only antibody like fibrinogen and thrombin that play the role in blood clotting. Another one, regulation, as I mentioned about hormone. This one is growth hormone. And blood glucose layer, as I mentioned, insulin and also glucagon. This one you will learn more detail in semester 2. This one is ceruloplasmin. This one you don't need to know lah. This one is more about biomedical part lah. Okay. And final one is storage. storage. So normally you can protein is also act as the storage. You can provide the energy lah. You can also provide energy to the human. And these are all the function overview of the protein function that I extract from the Campbell. You can make yourself. So I think that's all for today lah. That's all for this topic. So okay. So I already cover lipid and also protein. So any question? I think no. Yeah, normally is no. So easy, right? Very easy. So I already mentioned chapter 1 is very easy. It's just a revision because this all you already learn in SPM. You just do a revision only. So I think if no questions, so we will end today class. Now it's already 10:30. Okay. So next week we will cover back. So this Saturday no class. Next Saturday we will cover all the chapter 1 lah. We just left nucleic acid and also analytical technique. These two topic only. Next week we will cover it, and another week we will enter chapter 2. So I think that's all. If no questions, so we will see you next week. So thank you for your joining, and good night and have a good rest. Bye. 什么东西跌？刚才。